So they will can't do anything but fall to the bottom of the well from above. All together? I hope it makes sense to you. All right. So the end point, we're going to have to speed it up a little bit. The end point of the TODS ASO neutralization test is then the highest dilution of serum which shows no hemolysis or which shows bottoming. And we're going to be looking. So this is what bottoming looks like. Red cells fall to the bottom of the well, and the bottom of the well is curved, so they're going to fall together. So they're going to make a clump or a bottom, as opposed to lysis here. All right. So these are two controls that we would have done, your positive and your negative control. For your positive control, the titer, as we said, is going to be the highest dilution that shows bottoming or no hemolysis. So no hemolysis here, no hemolysis here, no hemolysis here, no hemolysis in 160, no hemolysis in 200. But when we get over to 300, we find hemolysis. So what would the titer be? 200, excellent. And then the titer for your negative control would be less than 40 in this case. All right? Good. What is it that you don't understand?
So you mentioned how you can test for both patients
each column represents a dilution. Each row represents a patient or a sample. So this right across here is your positive control, varying dilutions. This here is your negative control right across here. Dilutions of the control. So your positive control is going to be a sample that contains antibodies to streptolizin O. That's your positive control. Your negative control is going to be a sample that does not contain any antibodies to streptolizin O. That's right.
So, all affecting the entirety of that row is positive. However, the entirety of the first row.
Of the positive row?
Yeah.
No, it isn't. What does this indicate? What does this buttoning indicate?
It's positive, but we're diluting, which means that you're going to have some streptolizin, which is not all.
You're going to have some antibodies to streptolizin O, or okay, all right, you're going to have some streptolizin O, that's not bound.
Therefore cause the lysis, right?
But we're not, but we're not detecting streptolizin O. We know streptolizin O is there because we added it. We're detecting antibodies to streptolizin O. The antibodies bind the streptolizin O, and then when the red cells are added, the streptolizin O is neutralized and can't lyse the red cells. So hold on, let me tell you what the challenge I think you're having. We have antibodies in this sample here. Imagine that I have a vial here. Good. Now this vial, like I said, has antibodies. If I dilute that sample, I'm diluting the antibodies as well, right? So I want to get an indication of how much antibody is in the patient's sample, how ill the patient is.
And that would be, tell you whether they are recent or...
Quite so, yes. It will tell you whether it's a recent infection, whether it was long ago, because if it was a recent infection, you expect to have a higher titer. You expect to have a titer approaching 2400. As opposed to if it was an infection 10 years ago, you will have a lower titer, perhaps like a 160, a 60, or a 40, perhaps. So the titer gives you an indication of how ill your patient is, how long ago they had infection. I hope I'm opening some eyes now. All right, I'm happy. Thank you for the questions. Let us talk outside. All right. So how do we interpret your ASTA test now? We have all these results. We need to know whether or not the results are positive. So what we do is that we look at the, excuse me 1 minute, students. about that class. That was my department calling me, and I found it so strange that they were calling. All right. So we know the values. We can determine the titers. We need to know now what the titers mean. So the values are reported in Todd units, and the Todd units are simply a reciprocal of the dilution. So a negative astot would correspond to less than or 80, less than or equal to 80 Todd units, or less than or equal to 80 dilution. A doubtful is going to correspond to a titer of 160, and then a positive would be 200 and upwards. So greater than or equal to 200 would be considered a positive, and there you have it. So then, I think we can now do some interpretations of our tests. So this is your positive, we ran through it before. That's a titer of 200. Your negative, we have no lysis, we have no buttoning, we only have hemolysis, so that's a titer of less than 40. Good. What then is your titer for patient T1? I heard 160, I agree with you. This is the last dilution or the last well that has buttoning. Now then, what is your interpretation? Negative? Doubtful, excellent. How about patient T2? The titer is 300 and that's a positive. Patient T3, what's the titer? 60, and the interpretation is negative. How about patient T4? A titer of? Right, so this is a patient who had a very recent infection. Excellent, I love how you're getting it. So that's definitely a positive. Patient T5, 300, and so that is a, I think that's a 600, this is an error here, that's a 600, you see how I catch you? That's a 600, so that's a positive. And how about patient T6? So just like for your negative here, where we had no buttoning, and therefore no indication of positive, so we record that as a less than 40, saying that if we were to make a lower dilution, we don't know if it would have been positive. Now for your patient T6, we don't know if we continued with the dilutions, if we would have eventually achieved an endpoint, like a final endpoint where there's no more lysis. So we just say greater than 2400. There really is no need to continue with that dilution in any place, because that means that your patient has a very high titer. What you need to do now is start treating to get that titer down. Go ahead. Well, you just ended, she's asking what would you do if you ended up with a doubtful. If you end up with a doubtful, based on the patient's other signs and symptoms, did you isolate a group A strep from their sample? Was there a fever? Did they show any, was it a skin infection? Was it a scarlet fever? Based on all of that, which is why there's no one lab test that we use to diagnose. So you're going to use all the other criteria to help you to decide whether or not the patient has an ASTO. And really, if you're a positive ASTO or a positive anti-streptolysin O test, and really though, if your patient does have a positive streptolysin O test, anti-streptolysin O test, or if they've had a recent strep A, group A infection, unless there was an error in performing their test, there's no way they should come down as doubtful. Pardon me? Yes. Or someone explain to it. Remember this? That's what we're using. Make sense? I think we'll have to talk after class because we need to get moving. Alright? See me after. Alright, there are other tests that we can use to detect other antibodies that can be produced by Streptococcus group A as well. So it can, Streptococcus group A can also produce DNase, alright, DNase B. So if it produces DNase B, it means that the patient also produces antibodies to DNase B, and those are called anti-DNase B. You can also detect anti-hyaluronidase. You have supporting laboratory tests such as the CRP, C-reactive protein, and ESR, erythrocyte sedimentation rate, but these are non-specific indicators of inflammation. So if your patient had an infection with another organism, it won't differentiate between the group A strep and that other type of infection. It just tells you that the patient has an infection or there is some amount of inflammation. But of course, all these tests together can give you a good picture of what's happening. Now let's talk about infective endocarditis. This is an infection, of course, of the cards in the cardial surface, endocardial. We classified into 4 different groups, and we have them here. You would have gone through this, so I'm not going to spend very much time. Infective endocarditis can be either acute or subacute, but this depends on the severity as well as the progression of the disease. Infective endocarditis typically occurs in the heart valves, and you can see the different heart valves, but you can have the infection occurring at other sites of the heart as well. In the case of acute infective endocarditis, etiologies include Staphylococcus aureus, with which you're going to become very familiar by the time you finish med school. We have also Streptococcus groups A, B, C, and G, as well as Pseudomonas aeruginosa. There's also subacute infective endocarditis, and the difference between these two is simply that your acute is rapid onset, and your subacute is going to take a longer time for this to develop. So these now your subacute infective endocarditis tends to be caused by organisms that are less virulent than those ones that I mentioned before. So you have your Streptococcus viridans group, there's Staphylococcus epidermidis, Enterococcus faecalis, or Enterococcus group D strep. There's also Streptococcus bovis, or non-enterococcal group D strep. And then you also have what are termed as HASEC organisms, that is Haemophilus, Actinobacillus, Cardiobacterium, Eikenella, and Kingala, which is what you get the HASEC from. And you also have some other fastidious gram-negative organisms that may cause subacute infective endocarditis. Now, how are we going to diagnose this infection in the laboratory? We're going to start off with a specimen, which is up to you. You're going to collect the specimen and send to us. Some of the different specimens that you can use, there's blood, so we can do blood culture. You can collect valvular material as well as embolic tissue. These samples sound like postmortem samples, fortunately. You can also perform serological tests as well as molecular-based techniques. Now, typically when you're doing blood cultures, when you're collecting samples for blood cultures, you want to ensure that you collect the sample before you initiate treatment. Typically, it's about 3 to 6 sets, 3 minimum sets of blood cultures, and 1 set of blood culture bottles consists of 2 bottles, which is your aerobic bottle and your anaerobic bottle. That's a set, that's a complete set of blood culture bottles. Sometimes you may go other places, and based on resource constraints, you may only use 1, but ideally, 1 set should consist of 2 bottles. And of course, you want to collect your blood culture, your sample, before you initiate treatment. If you initiate treatment before collecting your sample, you're giving them antibiotics. These antibiotics might well be effective against the organism, pretty much kill it, so that when we do the test, we're not able to detect anything, or it becomes more difficult to detect it. In addition to that, you see here 3 separate sets, and that you're going to be timing them over a period of 24 to 48 hours. For your subacute infectious endocarditis, like I said before, it's a more slow onset. So based on how well your patient is, if you think you can delay treatment, then certainly you can delay treatment whilst you are collecting those samples. And then after you've collected the samples, then you initiate treatment. If it's a case of acute infective endocarditis, hey, you want to start treatment as soon as possible. So if you can even get the first one, before you start treatment, that is ideal. I think I saw a question. Oh, it's fine. Okay. All right. And if it's a bacterial endocarditis, the timing may not be so critical, so you can collect them over a shorter period of time. Now, when you're collecting, of course, you want to ensure that you're using an aseptic technique. You don't want to be introducing organisms into your sample, or worse yet, cause more infection for your patient. All right. So you use an aseptic technique. You're going to clean the skin with 70 to 80% alcohol, and then after that dries, 2% iodine. You're going to do that in a concentric fashion at the venipuncture site, and then allow that to dry, to air dry. In the meantime, you're going to disinfect the top of your blood culture bottles with alcohol, And I want to impress on you that you do not use iodine to clean the septa of your bacterial alert bottles. I'm going to show you the difference between your bacterial alert bottles and your manual bottles. What happens is that the iodine weakens the rubber portion of the bacterial alert bottles and makes it easier for them to become contaminated. So you clean with alcohol. For adults, this is important that you remember. In adults, you're going to be taking 8 to 10 mils of blood per bottle. All right, and in children, 3 to 6 mils of blood. And typically for your pediatric cases, it's usually just one bottle. 8 to 10 mils of blood per bottle for your adults. Now we have different types of bottles, and I'm going to be showing you different ones. Here you're looking at your green and pink bottles here. Remember, you disinfect with your alcohol, and then after you use a syringe to collect the blood, and then you transfer to the blood culture bottles. You're just going to inject it into the septum there, and you're going to mix very carefully, not vigorously because you might hemolize the blood, but mix very carefully, very thoroughly, so that the blood does not clot, right? Then you ensure that your samples are properly labeled. These bottles here that are green and purple, they're termed the bacterial alert bottles. We use them in an automated system in the laboratory, as opposed to manual bottles that sometimes we have to refer to. These green ones are not the standard ones that we use. These green ones have activated charcoal inside of them. So these ones are most suitable, for example, when a patient is already on antibiotics, because that activated charcoal here serves to bind the antibiotics, and so gives you a better chance of isolating the organism if it is indeed present. So you label your blood culture bottles, send them to the laboratory, and you remove the residual iodine from your patient's skin. Now we have the colors of the bottles distinguish them from the aerobic or anaerobic, from the pediatric or adult, as well as the presence of other substances such as resins or supplemented enrichment media. So these ones that I was showing you before, these are the ones that contain activated charcoal. So the green one in this case would be your aerobic bottle, and it's actually there, aerobic versus your anaerobic bottle. So your aerobic bottle is better suited to grow your aerobic organisms, and your anaerobic bottle for your anaerobic organisms. These are the standard bottles that we use in the laboratory. So your blue there is for your aerobes, and your purple for your anaerobic bottles, and these are the standard bottles. And the yellow bottle is your pediatric bottle, and of course that is also smaller than the regular ones. And as I said, a set of blood culture bottles consists of two bottles, an aerobic and an anaerobic bottle. Now here we are looking at the blood culture method when we're looking at when we're doing the automated method. All right, so once the blood culture bottles get to the lab, we place them into the automated instrument, this right here. It's called the Bacti Alert, and this instrument acts as both an incubator to incubate your blood culture bottles, as well as to detect whether there's any growth in the bottles. All right, so the instrument consistently monitors the bottles for the production of carbon dioxide, which is a byproduct of bacterial growth. And typically carbon dioxide can be detected within 13 to 20 hours of incubation. So here now, in our bottles, under normal circumstances, this is what your bottle looks like. The sensor, there's a sensor in the base of the bottle. So we have nutritious media. For example, we have brain heart infusion broth in the medium. When we add the blood there, and if there are any organisms there, we're providing a nice, nutritious and warm environment for them to grow. So if they are indeed present and they do grow, as they grow, they metabolize and they produce carbon dioxide. When the amount of carbon dioxide that they produce gets to a sufficient level, this is going to cause the sensor here to go dark. The color of the sensor is going to change to this dark color. Now, what's happening is that when you place your blood culture bottles on your analyzer, there's a laser that is passing through the base of each bottle at all times. And that laser there now, whenever the color of the sensor changes from that yellow color to that dark color, it blocks the sensor. And so the machine then detects that the laser is not, the light is not being detected passing through anymore. And then it sounds either a visible or an audible alert. Technologist on charge comes, they realize what's happening, they go to the computer which is attached to the machine, and then from there it tells them which bottle is given an alarm. They pull that bottle and they further process it. For your manual technique now, this is what your blood culture bottles look like. Not so fancy. but they do the job. See, anaerobic bottle contains bioglycolate broth, which would be the case for your Bacti-Alert, your automated bottles as well, and your aerobic bottle contains brain heart infusion broth. All right? Now, when your manual blood culture bottles are, when you collect them, inoculate them, label them, send them to the laboratory, we place them into the incubator for 24 hours before we start processing them. All right? So place them into the incubator for 24 hours, and then after that, every day or every 24-hour interval, we're going to visually observe these bottles for any sign of growth. Visual signs of growth include hemolysis, so the blood may become hemolyzed. We may see turbidity or cloudiness. We may see gas formation. There are some bottles that are especially made for detecting gas formation. We may have what is termed as pellicle formation. So pellicle formation now is like a skin or a thin layer of growth that is just on the surface. We may see that. Or sometimes we can observe visible colonies or puff balls within the media. If we observe that, we're going to pull those bottles and we're going to perform our processing. Now, on your automated blood culture bottles, as the machine is shooting that laser through your bottles, day 1, day 2, day 3, and it's not detecting anything, that incubation continues up until day 5. After day 5, the Bacti-Alert, which is automated, which is also connected to the LIS system, the laboratory information system, sends a message over to your LIS. It's been 5 days of incubation, not detecting any growth. And so the technologists will see that message there, pull that bottle. It's been 5 days, send a preliminary report, send a final report at this time, pardon me, and then send a final report to either to the ward or wherever it is that the patient came from to say that this culture is negative after 5 days or no growth after 5 days. In the case of your manual bottles now, we incubate for 7 days before we say no growth. So we incubate for 7 days, we're checking every day for signs of growth, and if we see no signs of growth, up until day 7, we are going to do a subculture. So we're going to take some of the fluid in the manual bottles, we're going to add that to a blood culture, a blood agar, in addition to other types of plates, blood agar, Mahanki, and so on. We're going to incubate those and observe for any sign of growth. If after this period of subculture there still is no sign of growth, at this point we set up our final culture to say no growth after 7 days. We're sure there was no organism present. If we do identify positive bottles though, we are going to isolate the organisms, we're going to identify them, and we're going to perform antimicrobial susceptibility tests to determine which antibiotic is going to be suitable for you to use to treat. All right, so usually we're using this agar here. It's called a Mueller-Hinton agar, or MH agar for short, Muellerhinton, 2 words, Mueller-Hinton agar or MH agar. We make up a suspension of the isolate that we identified. We swab it all over the surface of the agar so that we can get a confluent lawn of growth, a smooth lawn of growth, and then we add the antibiotic discs in this concentric fashion. Now, as soon as the antibiotic disc touches the agar plate, it starts to diffuse into the agar. The antibiotic starts to diffuse into the agar. Cover that, place it into the incubator, 18 to 24 hours, and then we're going to read. And what we're looking for are termed as zones of inhibition. So, from this point of growth here, straight through the center of the disc, all the way to the other side of growth, that is, we're measuring the radius, and we're going to compare that to our standards in the laboratory to determine whether or not the organism is susceptible or resistant to that particular antibiotic. So this looks like a good zone of inhibition. It's likely that this organism could be sensitive to this antibiotic, but in this case here, hey, that's a very small zone there. I would even say it's likely. It's obvious that organism is resistant to that particular antibiotic. So let's look at how we identify some common isolates that we'll obtain in our laboratory. Now we have Staph aureus, which we spoke about. This is what Staph aureus looks like, or Staphylococcus aureus. That's what it looks like on blood agar. And it's typically described as white or golden colonies on blood agar. If we do a gram stain, we obtain what are termed as gram-positive cocci in clusters. So they are gram-positive because they are purple upon gram staining, and they are cocci because they are round or spherical. Staphylococcus aureus typically presents in clusters, all right? So gram-positive cocci in clusters. So we see gram-positive cocci in clusters from our gram stain, and we found these white or golden colonies on the blood agar. I wonder what this could be. But first of all, simply because we saw gram-positive cocci, we performed a catalase test. Your catalase test differentiates your staphylococci from your streptococci. All staphylococci are catalase positive. All streptococci are catalase negative. All right, so we did our catalase test, and here we found that it's positive. All right, so we have our stuff. There are 3 clinically important staphylococcus species: Staph aureus, Staphylococcus. Anybody know the others? Epidermidis, Staphylococcus epidermidis, and Staphylococcus saprophyticus. Now, Staphylococcus aureus is the most important one. We're thinking of an acute endocarditis. All right, so we have a catalase positive, gram-positive cocci. We're thinking that we have staphylococci. We now need to identify this organism. We perform a coagulase test. This is a coagulase test, and we find that it is positive. So what we're looking for with the coagulase test is a clump, all right, a fibrin clot for the endpoint to show that your coagulase test is positive. And this organism was positive for coagulase. Ding, 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 ding. We've identified Staphylococcus aureus. What a bonus test is your novobiasin susceptibility, and Staphylococcus aureus is also susceptible to novobiasin.
Mr. Staphylococcus is catalase positive.
All staphylococci are catalase positive. You have 3 clinically significant species of staphylococci, but there are lots of staphylococcus species. The 3 clinically significant ones are aureus, epidermidis, and saprophyticus. So we've just described how to identify Staphylococcus aureus. All staphylococci are catalase positive. So when I get a gram-positive cocci from my gram stain that grew on my blood agar, I know that I have either staphylococci or streptococci. Those are the only 2 gram-positive cocci, aerobic gram-positive cocci, that you need to know right now. Good? So I cultured on my blood agar. These colonies grew. I did a gram stain from these colonies. I found that they were gram-positive cocci. I'm thinking either staph or strep. As a matter of fact, I'm thinking staph because it's gram-positive cocci in clusters, but I'm not sure entirely. I can't just go and jump and do those things. I must follow the method. After obtaining such a gram stain result, I performed the catalase test. My catalase test is positive. What does that tell me? It is a staph. No, it is a staph. I need to decide which staph species it is. It can be either aureus, epidermidis, or saprophyticus. I do my coagulase test. The coagulase test is positive, and Staphylococcus aureus is the only of these 3 that is coagulase positive. The other 2 are coagulase negative. If I want to identify the other 2, I have to do further tests. I saw a hand over here. Right? In addition to being coagulase positive, Staphylococcus aureus is also susceptible to novobiasin. For someone want to say something? All right. Now here's a group A strep that we've been talking about. Now group A streptococcus or Streptococcus pyogenes. So we cultured on our blood agar, and we're looking now at what is termed as a colonial morphology, simply what the organism looks like on media, on agar. They are described as beta hemolytic. All right. We do a gram stain, and this is what the gram stain showed. We're looking at gram-positive cocci in chains. So gram-positive cocci in chains, this sounds like strep. First of all, remember I told you, staph present in clusters. Streptococci tend to present in chains or in pairs, all right? So here we have chains. Plus they're beta hemolytic. This pattern of beta hemolysis looks strepish, but don't worry about that. We must follow the process. I can do that. Don't do that. We're gonna follow the process. So we have these beta hemolytic gram-positive cocci. They're gram-positive cocci. It can either be a staph or a strep. I must perform the catalase. Excellent. So I do the catalase, and the catalase comes up to be negative. And that means that I have a Streptococcus. Now, for your Streptococcus group A, there's a series of tests that we can do because there are other beta-hemolytic streptococci. But to make things simpler for you, we're just going to zoom into the bacitracin. Group A Streptococcus is susceptible to bacitracin. And here's my little trick that I used to do. Group A strep, sensitive to bacitracin. A, B. Simple, don't it? It's very simple because you will come to group B, which is C, and group D is E. So just wait until you get there. It will make more sense. You will see how simple it is. Next up is Pseudomonas aeruginosa. I can't believe you laugh at me. Now, Pseudomonas aeruginosa is a gram-negative bacilli, all right? You have your Enterobacteriaceae. Wait until you have all the tests, remember, you will see. Now you have what are termed as Enterobacteriaceae, which is a group of gram-negative bacilli, and typically most of them can be found in the gut, all right? However, Pseudomonas aeruginosa is a gram-negative bacilli, but it's not one of the members of the Enterobacteriaceae. Your Enterobacteriaceae are, for example, your E. coli, Klebsiella, and so on. Pseudomonas aeruginosa, you may hear it termed as a non-fermenter. Don't want to confuse it too much, that's a bit too much, but you'll get there. It produces this green pigment here, lovely green pigment. And I like to say it is one of the more pleasant smelling organisms in the laboratory. The rest can be pretty stinky, all right? It has a fruity, it's described as having a fruity or grape-like odor. I don't quite agree with that, but that's what the books say. It's more pleasant than the others is what I will say. But if you must describe it, it's a fruity or grape-like odor, all right? You can go on. Next up we have these green pigmented colonies on our agar, and our gram stain showed us gram-negative bacilli. We're going to perform an oxidase test. So your oxidase test is to your gram-negative bacilli what a catalase test is to your gram-positive cocci. All right, so when you have gram-positive test, when you have gram-positive cocci, you do your catalase. When you have gram-negative bacilli, you do your oxidase. Pseudomonas aeruginosa is oxidase positive. And then we perform a test that is termed the oxidative fermentative test. We simply want to determine whether the organism can utilize glucose oxidatively or fermentatively. It oxidizes glucose, but it does not ferment glucose. So we see that it is oxidative. The Streptococcus viridans now, again, gram-positive cocci, and remember I told you the strep, they are gram-positive cocci and they tend to appear in pairs or short chains. This one now is alpha hemolytic. You might not see the hemolysis very clearly here, but it's alpha hemolytic. And because we had gram-positive cocci, we did the catalase. Catalase is negative. Negative catalase tells us that we have Streptococcus. And so for your alpha hemolytic strep now, we do an optogen test. Now your Streptococcus viridans is resistant to optogen. And I just want to tell you now before we get there, Streptococcus viridans, I like to refer to it as the opposite of Streptococcus pneumoniae, because the Streptococcus pneumoniae now would have been susceptible to optogen. All the results would have been similar, except that it would have been susceptible to optogen. It's coming up, you're gonna see it. Enterococcus faecalis now, on blood agar, white colonies, they're described as white colonies, and again, gram-positive cocci in pairs and chains. Here you have your chains. And because we had gram-positive cocci, we did the catalase. We found the catalase to be negative, indicating to us that we had a strep. And so we're thinking, like I said before, I am just zooming in on the test that you would do to identify, but there's a host of tests that we would have done because we wouldn't have known what organism it is. So your Enterococcus faecalis, sometimes you would also hear it referred to as group D Streptococcus, is bile esculin positive. So remember I told you about group D, which is E. Now esculin here now, this is the English spelling. The American spelling drops the A. So you see why I said DE? Group D strep, bile esculin, DE. It's too simple, not true? I know that's why you find it so strange. I told you about Staphylococcus epidermidis. Here they are on blood agar, described as white colonies. When we do a gram stain, we find these gram-positive cocci. So because we have gram-positive cocci, we do our catalase, and we find our catalase is positive. That means that we have staphylococci. Now we have staphylococci. Which of the three could it be? We do the coagulase test. Uh-oh, the coagulase test is negative. And this means what? Excellent. It's not Staphylococcus aureus. It's either epidermidis or saprophyticus. We also do a novobiocin susceptibility, and we find that it is sensitive. Staphylococcus epidermidis is sensitive to novobiocin. Did we see any of that was sensitive to novobiocin before? Staph aureus, yes. Staph aureus is also susceptible to novobiocin. But we won't get confused because Staph aureus is coagulase positive, while Staph epidermidis is coagulase negative. All together? Great. Now we can have fungal causes of your infective endocarditis, and you'll hear about Candida albicans. Morphology, white or creamy colonies on your blood agar. We can do a gram stain on them, and we find that they show large gram-positive ovoid bodies, and they are gram-positive and ovoid, and they are much larger than cocci. So you won't mix them up with bacteria. All right, this is straight up yeast. We perform what is termed as a germ tube test. So what we do is we take up a bit of the growth, we inoculate that into a tube containing about a mil, less than that, maybe about 300-400 microliters of serum, serum, use plasma, and then we incubate that overnight. After incubation, we take a sample of that fluid there that would have contained the growth of the yeast, place that onto a microscope slide, and a cover slip, and we observe. Now, what we're observing for is the formation of germ tubes. Now, a germ tube is simply a hyphal outgrowth from the yeast cell itself. Now, for a positive germ tube, you should have no constriction between the hyphal outgrowth and the yeast cell. And to make it easy, it's 48, I thought it was 59. So this here is your outgrowth here, and there is no constriction. So this is a positive here, positive germ tube test. Once this here, you can see the constriction. That would be a negative test there. You can also have Aspergillus species, which this is a mold now. Fast growing colonies, maybe white, yellow, brown, and we can identify these based on performing a tease mount preparation. So what would occur is that we have the growth here on our petri dish. We take up a portion of that, place it onto a side in lactophenol cotton blue, so it stains the cells of the yeast, and we gently, ever so gently, tease it apart, add a cover slip, and observe under the microscope. So what we're looking for are the structures that tell us that, hey, which fungus this could be. And how these structures here look, these represent what your Aspergillus species look like. All right? And finally, septicemia shock. Sepsis, extremely complex reaction, and this is typically evoked to an infection caused by microbial agents. So our microbiological investigations help you, the clinician, to confirm that the patient indeed has sepsis and what is causing that sepsis. Specimens that we're going to collect, blood cultures, we can culture other fluids such as urine. Is it a wound that has caused this sepsis? So we can do wound swabs. We can also culture from other sterile body fluids, as the patient's symptoms would indicate, such as spinal fluid, joint or pleural fluid, and is it a recent intramuscular device that was implanted that is giving rise to sepsis? We have our universal containers, for example, if you're collecting like tissue material, biopsy or so. If it's a swab that you're collecting, you place it into your storage transport medium. And of course, this is what the commercially prepared storage transport media looks like. And if you're collecting blood or fluids, you place them into your blood culture bottles. Specimen, how we process the specimen in the lab, we start off with direct microscopy, which is gram stain. So we're going to do a gram stain directly on the patient's specimen, and then from there we'll also culture. So the gram stain will tell us what kinds of media to use based on what we observe. Is it gram-positive cocci? Is it clusters? Is it chains? Is it pairs? Is it gram-negative bacilli? That kind of thing. We incubate, we inoculate 2 blood agars. One we're going to incubate aerobically, the other we incubate anaerobically, and then we also inoculate a McConkey's agar. Most pathogens will grow well on your blood agar, and for your McConkey's agar, this helps to identify the different members of your Enterobacteriaceae. Remember I told you about the E. coli or Klebsiella? Simply we're going to divide them. Are they lactose fermenters or are they non-lactose fermenters? Lactose fermenters on McConkey will turn pink. Non-lactose fermenters on McConkey will turn a pale yellow or straw color, like urine. That's what your non-lactose fermenters will look like. And when I say lactose or non-lactose, lactose is incorporated into the media. So if the organism is able to ferment the lactose, it turns the media as well as the colonies. If it does not, it turns the media as well as the colonies a pale yellow or straw color. What are some of the organisms implicated in sepsis? Gram-positives, your Staph aureus. Can't get enough of your Staph aureus. Staph epidermidis, Enterococcus, also Streptococcus pneumoniae. And some your gram-negative organisms, they have E. coli, Klebsiella, which I've spoken about, Pseudomonas species as well as fungi and anaerobic organisms. Told you about, I briefly mentioned Streptococcus pneumoniae when I told you about Streptococcus viridans. Here it is now. Gram-positive cocci in what are termed as lancet pairs. Lancet pairs almost elliptical, all right? So they're joined end to end like this, and they're lancets. So they are termed because they look somewhat like the blade, a surgeon's blade, so they have that curve there, right? On blood agar, they will demonstrate alpha hemolysis. Of course, they are catalase negative because they are streptococci, and here they are susceptible to optogene. So here's your optogene disc, and you can see that zone of inhibition there. Good. With your viridans, there was no zone of inhibition. Here's your E. coli, and whenever you hear the term flat, dry lactose fermenters, your go-to food is a term I want to use that is slipping me right now. Your default is E. coli. Whenever you hear the term flat, dry lactose fermenters, you default to E. coli. All right? On gram stain, gram-negative bacilli. Now remember I told you for your gram-negative bacilli, we perform oxidase. Here it is negative. Remember what was the result for your Pseudomonas? Oxidase positive, right? So Pseudomonas oxidase positive. They're telling you that it's a different kettle, it's a different group of organisms than your Enterobacteriaceae. And then don't worry about these from Figler's all the way down to lysine. These are termed as biochemical tests, and we use them in the laboratory to help us identify the different members of the Enterobacteriaceae. Based on the results that we obtain, those pattern of results can tell you which organism it is. And of course, lactose fermenters, because here on McConkey's agar, they are pink. Here now are your Klebsiella pneumoniae. So whereas E. coli is flat, dry lactose fermenters, your Klebsiella is mucoid raised lactose fermenters. Pink on your McConkey's agar, and you can see that they look a little bit more shiny. They are also raised. So mucoid raised lactose fermenters, also gram-negative bacilli, also oxidase negative, and the different pattern of your biochemical tests will identify your Klebsiella pneumoniae, also your Enterobacteriaceae, and that is a big factor.